Hello, everyone, and welcome to today's webinar, The Latest Developments for Enriching and Analyzing Antigen and Virus-Specific T-Cells. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I'd like to now welcome our speaker, Lorenz Fülle, the Global Product Manager for Antigen and Virus-Specific T-Cells at Miltenyi Biotec. Lorenz, you may now begin your presentation. Welcome.
And welcome again to today's webinar on our latest developments for enriching and analyzing antigen and virus-specific T-cells. I'm Lorenz, and I'm a global product manager here at Miltenyi. And in today's webinar, I will introduce to you our complete workflow, so from sample preparation to cell analysis, to work with this very rare, but at the same time also very important T-cells. So also in regard to the experimental workflow, we at Miltenyi Biotec strongly believe that the future of T-cell analysis has to be more standardized to generate more robust and also comparable results. So enjoy. Before I dive really deep into the topic, I want to quickly highlight a new product, the SARS-CoV-2 T-cell analysis kits that were released in December last year. These kits enable an efficient stimulation and sensitive detection of SARS-CoV-2 reactive T-cells in human PBMC samples. The presence of these SARS-CoV-2 reactive T-cells in donor samples then indicate an exposure of the donor to the SARS-CoV-2 virus. And the kits are really designed to be an all-in-one solution, and they contain everything. So antibodies, stimulation reagents, controls, buffers, and also a protocol. So if you want to know more about the kits, you have to stay tuned. So here you can see today's agenda. So I will firstly discuss the important role of T-cells in COVID-19 and also more general viral infections, and then also talk about some strategies on how to best analyze them. Next, I will talk about some of the key challenges when working with these rare T-cells, and also how Miltenyi and our products can help you overcome those challenges. I will end with a short look beyond T-cells in virus research and present our new recombinant SARS-CoV-2 proteins that can be used, for example, for the analysis of SARS-CoV-2 specific B cells. So COVID-19 has been really an unprecedented global challenge. And we think only together we can defeat COVID-19 and return to a, let's say, more normal life. So just as a quick repetition, SARS-CoV-2 is the virus that causes the respiratory illness COVID-19. And still, SARS-CoV-2 still exhibits very high infection rates and also numbers of cases worldwide. In general, COVID-19 has a very broad spectrum of disease severity and symptoms. And the symptoms range from asymptomatic or mild to really life-threatening illness, especially, of course, in groups of high risk. And the control measures, such as lockdowns or social distancing, had and still have very severe social and economic consequences. And this is exactly why we need more tools for an efficient and precise diagnosis of COVID-19 to then hopefully boost the development of more effective therapies. And as Dr. Anthony Fauci, the chief virologist of the US government, recently said on a Keystone Symposia, unprecedented progress has been made, but much more needs to be done. So consequently, also Miltenyi Biotec will do its best to support your virus-specific T-cell research. So there are also some good news. And this is that effective vaccines have been approved by various regulatory agencies, and they're also currently being rolled out. So while all of these approved vaccines are more or less really high effective, I've brought you just some examples to demonstrate the effectiveness of some of these vaccines. So for example, here on the left, you can see the Pfizer-BioNTech vaccine and also the Moderna vaccine in the middle. And they actually show a protection of up to 95% in vaccinated people. And here on the right, I've selected an important publication from the so-called Oxford COVID vaccine trial group that analyzed immune responses in people that received the vaccine from AstraZeneca. And as you can see, vaccination actually induced both. So a very strong antibody response as well as a strong T-cell response. So now you might ask, why do we actually need to study T-cells, especially in COVID-19, in more detail? Well, first of all, of course, because we simply need a more accurate picture of SARS-CoV-2 immune responses. And just as a note, I really have to say, for me, it is absolutely amazing how many studies are currently being conducted to disentangle the immune responses to SARS-CoV-2. But let's have a look at some of the key findings. So for example, it has been shown 10 to 30% of patients do not produce detectable levels of SARS-CoV-2 specific antibodies. And furthermore, in patients with asymptomatic or mild disease, the antibody titers are actually really low and also rapidly decline. In contrast, it has been shown that SARS-CoV-2 reactive T cells are generated even in the absence of virus-specific antibodies. So, for example, in this figure from a study by Secretion et al. Published in 2020, you can see that T-cell responses were detected more frequently compared to virus-specific antibodies, particularly in cases of mild infection, shown here in red and in this greenish. Also importantly, the analysis of SARS-CoV-2 reactive T cells can also be used to monitor long-term immunity, be it after natural infection or also after vaccination. And also particularly in regard to vaccines, it will be very important to study the implications of cross-reactive T cells. So these are T cells that were originally generated against coronaviruses, for example, for the design and analysis of all the upcoming trials that investigate COVID-19 vaccines. And last but not least, also SARS-CoV-2 reactive T cells can be generated and also used as a treatment option, for example, in passive immunization. And down here on the bottom, you can also click on these selected references that I picked here. So why are especially T cells so fundamental for an efficient antivirus immune response in general? So apart from the innate immune system and the B-cell mediated antibody responses, T cells really play a central role in the organism's antivirus defense. And of course, all these parts, so innate immune responses, ruminant immune responses, are strongly interdependent and also need to work together efficiently to generate antiviral immune responses. However, I just want to point out that only assessing antibody titers after infection will not result in the full picture. And this is exactly why we need to have a look at virus-specific T-cell responses. So why are virus-specific T-cells so fundamental for the immune response? Well, of course, they not only kill virus-infected cells directly, but also support B-cell responses. And most importantly, virus-specific T-cells provide long-term immunity and are therefore relevant for favorable disease progress and also to have a better prognosis of disease. So now we can have a look at how to best analyze these antigen and virus-specific T cells. Well, generally, the methods can be classified into two main categories. So first, there are the direct analysis methods where we analyze T cell specificity with MHC multimers, for example. And here, the T cell receptor is labeled with a peptide MHC multimer complex to directly identify antigen or virus-specific T cells. And although these methods certainly have a very high specificity, they also come with some limitations. For example, they work really good for CD8 positive T cells, but only very limited for CD4 positive T cells. There's also no info on the functional capacity or the T cell subset, and it is also limited to define peptides, let's say antigenic epitopes and MHC molecules. And then second, there are the indirect analysis methods. And here, more functional parameters, such as, for example, the expression of certain activation markers or cytokines, but also, for example, cytotoxicity or proliferation can be analyzed. So you have to understand that these are indirect readouts for T cells that were stimulated, for example, with a specific peptide antigen. Suitable activation markers that could be analyzed are, for example, CD69, CD137, or CD154, or also cytokines like interleukin-2, TNF-alpha, or gamma interferon. So these methods have the advantage that they really work well for all T cell subsets, also including regulatory T cells. They provide T cell subset specific information and are also completely independent of the used antigen or the MHC status of the donor. Most importantly, however, the biggest advantage of these indirect methods is that all relevant information can be obtained using flow cytometry-based assays. So for today's webinar, I will focus on these indirect analysis methods. But let's first have a quick look into the key challenges that arise when working with these rare cells and how we can help you to overcome them. So as briefly mentioned before, the low frequency of these rare antigen and virus-specific T cells is definitely the biggest challenge for a reliable analysis. All the more, it is really important to optimally prepare the starting material without losing any of these precious cells. Furthermore, to ensure higher cell numbers, the cells can also be pre-enriched magnetically or via flow sorting. Another common problem is insufficient stimulation. And only by using really powerful reagents and also optimized protocols, the cells will be efficiently stimulated. Furthermore, the analysis is often hampered by high background staining. And this is mostly just because the cells are just so rare. And here, reagents for specific labeling and also really sensitive detection methods can help to lower the background. And finally, there's often a rather slow translation of these findings into the clinic. And here, Miltenyi offers a large portfolio for translational research that could also help you to speed up this process. So to efficiently isolate, stimulate, stain, and analyze antigen and virus-specific T cells, we combine different technologies over an entire workflow to ensure successful experiments. Yeah, and of course, we think that the right product combination here is the key to success. So now let's have a quick look on the workflow that can be applied for the analysis of antigen and virus-specific T cells. So first, the T cells need to be isolated from a donor sample, and this could be blood or tissue. For tissue dissociation, of course, you can use our GentleMax tissue dissociator, which is a really good choice. Please note that for the analysis of antigen and virus-specific T cells, you also need antigen-presenting cells, because they efficiently prime and activate the T cells with a certain antigen. When working with PBMCs, for example, this is already the case. However, with isolated T cells from tissues, antigen-presenting cells have to be added separately to the culture. Next, the cells need to be efficiently stimulated with the antigen of interest, for example, using our large portfolio of SARS-CoV-2 Peptivator peptide pools. Following the stimulation, the antigen-specific T cells will get activated and upregulate certain activation markers and also start to secrete cytokines. The cells can then be stained intra and extracellularly to assess their phenotype and also analyze cytokine expression. Before the analysis, these rare antigen-specific T cells that were activated can also be pre-enriched magnetically. And finally, the cells will then be analyzed, for example, via flow cytometry or further sorted on a specific cell sorter. And you can find a very good overview of the whole antigen and virus-specific T cell workflows, including protocols and other product solutions on our application websites by following these links here in the bottom. So now we will quickly go through the different steps of this workflow. And of course, successful experiments, and you probably better know this than me, always start with efficient sample preparation. And one of the most widely used sources for antigen and virus-specific T cells are still PBMCs, because they actually contain both the antigen-specific T cells as well as the antigen-presenting cells. And thereby, PBMCs actually provide, let's say, functional immunological context for the immediate analysis of antigen-specific T cell responses. So usually, PBMCs are prepared via density gradient centrifugation. However, when working with, for example, contaminated samples, somehow compromised samples, very small samples, or very rare cells, density gradient centrifugation can be very tedious, inefficient, and in some cases even dangerous. So, and this is where our MACSPrep PBMC isolation kit comes in. The MACSPrep PBMC isolation kit provides a very fast and untouched, this is very important, untouched isolation of highly pure PBMCs with a high recovery. The kit is optimized for small blood samples of 1 to 10 ml and does not require lysis of red blood cells or even density gradient centrifugation. The isolated cells are perfectly suited for downstream analysis, and you can even process multiple samples in parallel. On the graph here on the side, you can see that the recovery of various immune cell subtypes within the PBMCs is at least as good, if not even better, with the MACSPrep PBMC isolation kit, at least when compared to density gradient centrifugation using FICO. So the kit really eliminates any user-dependent inconsistencies and performs reliably for every separation. And this in turn will then help you to further standardize your T-cell assays. Again, you can also follow the links behind this yellow circles here on the top to find more information. So now, after preparing the cells, for example, from PBMCs, the next crucial step in the workflow is this antigen-specific stimulation in vitro. And here, the most efficient antigen-specific stimulation of T cells can be achieved with our Tivator peptide pools. These Tivator peptide pools efficiently stimulate both CD4 as well as CD8 positive T cells, and we cover an extensive portfolio of virus, tumor, microbiota, and even autoantigens. The peptide pools always consist of 50-mer peptides with 11 amino acid overlaps, which cover, let's say, the complete sequence of the studied antigen. And our Tivators are available in research, premium, and even in max GMP grade, which will allow a rapid translation into the clinic. And finally, they're also available in a convenient 96-well format for high throughput applications. So we think our Tivators can really help you to standardize your T-cell assays further and are the ideal stimulation reagents for antigen and virus-specific T cells. So most notably, we have many different SARS-CoV-2 Tivators, and the portfolio, due to the current situation, is growing very fast. So for example, we recently added the so-called SARS-CoV-2 Tivator Select, which contains immunodominant epitopes of the whole virus proteome. So it's actually a pool of 88 structural and non-structural SARS-CoV-2 peptides. Importantly, SARS-CoV-2 Select is already available in both premium as well as in max GMP grades. Of course, we also have other SARS-CoV-2 Tivators which cover different parts of the spike protein, for example, as you can see here on the right. However, we also have specific nucleoproteins and also membrane protein Tivators. And as for the whole portfolio, we will continue to develop new Tivators, for example, for the new SARS-CoV-2 mutation variants that currently arise from the UK or also South Africa. And you can also follow the links here to get a more detailed overview on the Tivator website. Also watch a very nice video that explains the technology in more detail. And yeah, we also generated some exemplary data using the SARS-CoV-2 Tivators. And here we can see that stimulation of T cells results in efficient activation. So in this example, we used blood samples of healthy donors and also a COVID-19 survivor. They were lysed with RBC lysis buffer and then stimulated for 4 hours with the respective Tivator or left untreated. The cells were then stained using an optimized intracellular staining protocol and our SARS-CoV-2 T cell flow panel. And here, antigen-specific activation was assessed by analyzing interferon gamma production by CD3 positive cells, and the plots clearly show that COVID-19 survivors present elevated levels of SARS-CoV-2 reactive T cells following the stimulation with one of the Tivators. So, of course, every stimulation experiment requires a reliable positive control. And here our CytoStim reagent can further help to standardize your T cell assays. CytoStim has been specifically developed for the rapid and efficient stimulation of human T cells. It activates T cells by binding to the TCR and cross-linking it with the MHC molecules present on antigen-presenting cells. It is an antibody-based reagent and can be considered as a non-toxic alternative to the super antigen SEB. Here in the graph on the side, you can see that PBMCs can be efficiently stimulated using cytostim. And also upon stimulation with cytostim and in the presence of antigen-presenting cells, CD4 and CD8 positive T cells start to secrete cytokines such as interferon gamma, TNF-alpha, interleukin-2, and will upregulate activation markers within a few hours. So cytostim really is the perfect positive control for stimulation of antigen and virus-specific T cells. So following this stimulation, we will now take a look at tools that can be used for pre-enrichment of antigen and virus-specific T cells. So, of course, still, flow cytometry is the most widely used method for the analysis of rare cells in general. However, even established methods like flow cytometry have their limits and also challenges, especially as antigen and virus-specific T cells are so extremely rare. And this was already illustrated years ago in a very nice publication by Bacher and colleagues, where they say that despite its high sensitivity, standard flow cytometry is always limited by the number of events that can be acquired, as well as the staining background, which is typically between 0.01 and 0.1%. So these limits really restrict the analysis of cell populations with such low frequencies. So although in most instances flow cytometers are actually sensitive enough to detect at least some cells, there might be certain conditions where hardly any specific T cell can be detected. And here you can use our MicroBeads for pre-enrichment. Moreover, to keep this background staining at an absolute minimum and enable a high resolution, you can use our specific labeling reagents coupled to very bright fluorophores. And here our recombinant reaffinity antibodies really provide high specificity, purity, and lot-to-lot consistency. Furthermore, the recombinant design actually completely eliminates the need for Fc receptor blocking. And further, our really bright Vio dyes can also enhance the detection capabilities of these rare cells. So we think that especially for this rare cell analysis, enrichment can be the key for successful analysis. And then let me show you some examples. And here, the results after pre-enrichment using our CD154 or CD137 MicroBead kits are really amazing. So both markers are actually upregulated on CD4 positive and CD8 positive T cells after antigen stimulation, and are therefore really suited for the pre-enrichment. So in this example, PBMCs were stimulated with the CMV Peptivator, or also left untreated as a control. Then the antigen-specific T cells were either enriched using the CD154 MicroBead kit for CD4 positive T cells, or the CD137 MicroBead kit for CD8 positive T cells. The cells were then counterstained for CD4 and CD8. And as expected, hardly any cell expressing CD154 or CD137 were found in the unstimulated sample, independent of enrichment. However, following the stimulation, the antigen-specific T cells heavily upregulated the respective markers and could therefore also be easily enriched with the respective MicroBead kits. These isolated cells can then be used for any downstream applications, like, for example, sequencing of T cell receptors. And if you want to have a look at our nice tech paper here on the top, this really nicely illustrates the technology in much more detail. So in that sense of pre-enrichment, I would also like to introduce the so-called Rapid ARTE protocol, which is a really good solution for the analysis of rare and antigen virus-specific T cells. So Rapid ARTE combines cell stimulation, surface marker staining, intracellular cytokine staining with an enrichment step based on, for example, CD154, which, as we just saw, is a really good marker to enrich rare CD4 positive T cells. Strikingly, however, permeabilization and intracellular cytokine staining can be directly performed in our MAX columns during the enrichment. These so enriched cells and stained cells can then be eluted and easily analyzed on standard flow cytometers, like, for example, on our MACSQuant analyzers. Of course, you can also modify Rapid ARTE for example, CD8 positive T cells by using CD137 or any other markers for enrichment. So in conclusion, Rapid ARTE is not only very fast, so it only has it's only a 1-hour protocol with 30 minutes hands-on time, but it's also extremely sensitive due to this enrichment step. So now we can also have a look at a virus-specific example where the Rapid ARTE protocol was applied. So here, PBMCs from a COVID-19 survivor or healthy donor were either stimulated with a mix of SARS-CoV-2 peptivators, or in this case, an influenza peptide mix as a control, or left untreated. And as seen in the middle panel, the rare SARS-CoV-2 reactive CD4 positive T cells could be successfully enriched via CD154 and then easily visualized using flow cytometry. And the data were provided with the courtesy of Alexander Scheffel and Petra Bacher from the University Clinic in Schleswig-Holstein in Kiel, Germany. And you can also have a look at their publications listed here on the side to find more details about the enrichment of antigen-specific T cells. Furthermore, I also want to point out a cell separation product that is absolutely unique, and these are the so-called cytokine secretion assays. So cytokine secretion assays, or CSA, allow the viable enrichment of cytokine secreting cells from both mouse and human samples. And the principle is actually rather simple. So first, the secreted cytokines are immobilized on the cellular surface by the so-called CAT reagent. Basically, thereby the cell labels itself with its own cytokines. In the second step, the immobilized cytokines are then labeled with fluorophore-conjugated antibodies called detection reagents. And with that, living cells can then be detected without intracellular staining using flow cytometry. In an optional third step, the fluorophore-labeled cytokine-secreting cells can then be specifically enriched and used for downstream assays with the help of anti-fluorophore magnetic beads. So consequently, our cytokine secretion assays come in two versions. For flow analysis only, we offer the various detection kits, while for the enrichment and analysis of viable cytokine secreting cells, we have several enrichment and detection kits. So of course, we also generated some exemplary data with the CSA assays. And as you can see, they work very efficiently to enrich, in this case, viable virus-specific T cells based on cytokine secretion. And also here, we use PBMCs of a CMV positive donor, stimulated those PBMCs for 4 hours, either with CMV lysate or a CMV peptivator. The responding cells were then isolated according to the secretion of TNF-alpha or gamma interferon using the respective detection kits. And as you can see in the stimulated samples, a distinct population of cytokine secreting CD4 and CD8 positive T cells were specifically enriched. Whereas in the unstimulated control sample, no cytokine secreting cells could be detected after the enrichment. Although in some cases we strongly recommend to enrich the cells before analysis, you can often also go ahead and stain the cells for flow analysis immediately after the stimulation. And we will have a look at our products for flow staining now. So as already mentioned before, we have the reaffinity antibodies and VIO-dyes. And I really have to say they have been designed with flow cytometry in mind. So the reaffinity antibodies are recombinantly engineered. What does this actually mean? So simply, it means that we have a full control over their genetic sequence, and we can express them in mammalian non-hybridoma cell systems. And therefore, they have a higher purity and also lot-to-lot consistency compared to conventional monoclonal hybridoma antibodies. Furthermore, they provide a really low background staining, which makes them really good tools for especially rare cell analysis. Our VIO-dyes, on the other hand, are our own fluorescent dyes. And I have to say they work really well in conjunction with the reaffinity antibodies. They have a very high stain index and also low spillover, which make them a really good tool for this clear discrimination of these very rare cells, especially when you have a high background. So, and here we just listed several reaffinity antibodies that could be of interest when analyzing antigen and virus-specific T cells. So you can join this, let's say, flow revolution and have a look at our reaffinity antibodies that were really developed, I have to say it again, with flow cytometry in mind. And of course, we are also very happy to help you designing your own customized panel. So when it comes to flow analysis, we of course do not only have the reaffinity antibodies and the VioDyes, but we also have ready-to-use optimized flow kits. And we'll have a look at those now. So now let's just take another look at our new SARS-CoV-2 T-cell analysis kit, PBMC Human, that I already shortly introduced to you in the beginning. The kit was specifically developed for the efficient stimulation and precise analysis of SARS-CoV-directed T-cells in human PBMC samples, and is currently for research use only. And the kit is really an all-in-one solution. It includes the SARS-CoV-2 peptide pool for antigen-specific stimulation, and also cytostim as a positive control. It includes all 8 flow antibodies and live data that were carefully selected and optimized to enable a precise analysis of the SARS-CoV-directed CD4 and CD8 positive T cells. And we also intentionally left one position free to include your own antibody of choice. The included stimulation and staining protocol was specifically developed for the kit. And of course, the kit also contains all required buffers, reagents, such as Brefilin A, and of course has a very high capacity of 100 tests for up to 10 to the power of 8 cells. So here on the top, you can see the workflow of the kit. So after isolation and stimulation of PBMCs with the included SARS-CoV-2 Peptivator, the cells are stained with our specifically optimized flow panel shown here in the table, and then analyzed using standard flow cytometers like, for example, our MACSQuant analyzers. And it's also good to know that you can also create your own stimulation mix by combining different Peptivators. Right now, the kit is designated for PBMC samples only. However, we will soon release additional versions, for example, a whole blood kit or kits with other Peptivators. And as you can see here in the plots on the side, these specifically activated SARS-CoV-directed CD4 and CD8 positive T cells can be both detected in just one sample through the combined analysis of various activation markers, like, for example, CD154 or cytokines such as gamma interferon or TNF-alpha. Of course, we've also generated some data with this kit, and in this nice example, we stained, again, PBMCs of SARS-CoV-2 reactive donors after stimulation with the indicated Peptivator, and then stained them with the panel that is included in the SARS-CoV-2 T-cell analysis kit, plus CD69 in this case. And here we used a mix of CMV and EBV Peptivators as a reactive control. So for analysis, we pre-gated then on CD4 or CD8, and then assessed CD154 and TNF-alpha expression on CD4 positive T cells and CD69 and gamma interferon on CD8 positive T cells. And as you can see, T cells respond specifically to the stimulation with the Peptivators and then indicate exposure of the donor to the SARS-CoV-2 virus. So we do not only have the recombinant reaffinity antibodies or the SARS-CoV-2 T-cell analysis kit, but we also have other really convenient, ready-to-use flow analysis kits. For example, the Rapid Cytokine inspectors, which are also true all-in-one kits that allow a rapid multi-parameter analysis of both activated CD4 positive and CD8 positive T cells through a combination of extracellular staining and intracellular staining of various cytokines. We also have the so-called MACSPlex cytokine kits, which are available for both human and mouse samples. And in these kits, we use multiplex analysis to measure concentrations of up to 12 or 10 different secreted cytokines in cell culture supernatants. The technology is based on our MACSPlex capture beads that work with basically any standard flow cytometer. And of course, the MACSPlex portfolio is continuously growing. However, we already have a very large range of different kits, such as, for example, for the analysis of exosomes or cytotoxic T cells. And then last but not least, we also have the so-called 8-color immunophenotyping kit, which enables an optimized flow-based evaluation of various leukocyte subsets, such as, for example, monocytes, neutrophils, eosinophils, TMB cells, also NK cell populations. And of course, all of these kits are compatible with our very fast and convenient express modes that are available on all of our MACSQuant analyzers. And you can imagine these express modes as some kind of software module that really help you to automate flow analysis. And this will not only speed up the whole analysis process, but also help you again to further standardize your assays, thanks to, for example, automated gating or predefined analysis templates. If you are interested in the express modes or the MACSPlex kits, you can have a look at our very nice other webinars by following the respective links here on the site. So finally, we will now also take a short look at our own very powerful cell sorter, the so-called MACSQuant Tyto. So the MicroBead Tyto also really revolutionizes cell sorting by offering some unique advantages. It is definitely the ideal sorter for infectious samples, as in virus research operator safety has to come first. The Tyto really enables safe cell sorting, as the sample stays inside a closed cartridge system for the entire aerosol-free sorting process. Furthermore, the Tyto is also really gentle to your cells. It only uses very low pressure and does not apply any charge to your cells. And this leaves the Tyto sorted cells, so to say, in a really good condition to be used for any downstream applications. And also the full sterility of the sorting process make the Tyto really ideal in the context of translational applications, especially in combination with our Max GMP consumables. So here the Tyto is already part of several clinical studies involving, for example, engineered immune cells or also CAR T cells. And another key advantage of the Tyto is its small size, like all of our MACSQuant analyzers. And this actually makes it possible to directly place them, for example, in a flow cabinet. And let me just briefly point out that several Tytos are also being used in the context of SARS-CoV-2 research already. So here, for example, for the isolation of SARS-CoV-2 specific T cells, also SARS-CoV-2 specific B cells. So we think, and we also know, that our Tyto is a really good solution for gentle sorting of rare cells from, of course, not only, but also infectious material. So finally, here you can see a nice schematic overview of our whole antigen and virus-specific T-cell portfolio and the workflow. And as you can see, we actually cover everything. So from sample preparation to cell culture, cell simulation, cell separation, and also cell analysis. And again, if you want to have a closer look, you can check out the respective application websites for antigen and virus-specific T cells. You can watch our other webinars or also check out the large T cell research brochure. So now you might want to ask, is it really all about T cells in virus research? No, definitely not. So of course, B cells, NK cells, and myeloid cells also play a central role in viral infections in general. And of course, here, Miltenyi also offers really nice solutions for your virus research regarding these other cell types. So you can follow the links to reach the respective application page, but I would also like to quickly show you some of our highlight products that we have for B cell research. So for example, we offer a large portfolio of recombinant SARS-CoV-2 proteins, such as, for example, different variants of the spike protein, the nucleoprotein, or even the envelope protein. And these proteins can then, for example, be used for the analysis and isolation of SARS-CoV-2 specific B cells. However, using our bead-based immunoassays, you can also use these recombinant proteins to detect SARS-CoV-2 specific antibodies. So, and here you can see an exemplary workflow that could be applied to isolate SARS-CoV-2 specific B cells, for example, for the generation of monoclonal antibodies. So first, the B cells would be isolated from SARS-CoV-2 positive individual using, for example, our CD19 MicroBead. So the cells would then be either be stained with only flow antibodies, or also tetramers that were generated with the SARS-CoV-2 recombinant proteins. And the cells could then be directly phenotyped, for example, using flow cytometry, or also further expanded and also enriched for various downstream applications, such as, for example, single cell sequencing or B-cell receptor cloning. And finally, this could then be used to generate new cell lines for the production of monoclonal antibodies that could be used as a treatment or in other applications. So of course, this was very brief, and you can find more information by following these links. Also check out our very new poster where we analyzed both the humoral and cellular responses, so B cells and T cells, to SARS-CoV-2 in COVID-19 convalescent donors. And of course, we'll have a very specific webinar addressing only this topic coming up very soon. So to sum up, the most important message here is that we will manage the crisis together. And there are a lot of benefits that are coming from a comprehensive virus-specific T and B cell research. For example, we can clarify highly relevant questions about reinfections, vaccines, protective immunity, also the role of cross-reactive immune cells. We can use this analysis of these cells to monitor long-term protective immunity, be it after natural infection or vaccination. And of course, this will then hopefully lead to a better understanding of the antiviral immune responses in general, better disease prognosis, better treatment options, and then hopefully also have a better planning certainty and a faster return to a more habitual or normal life, to say. I really hope that by now I could convince you that Miltenyi covers the complete antigen and virus-specific T and B cell workflows to support your research. So before we end, please just acknowledge the displayed disclaimer. And then with this, I would like to close with this very nice picture of our company campus in Bergisch Gladbach. And of course, you can always visit our website to find more information on all the mentioned workflows and products. And yeah, I'm really thankful for your attention.
Thank you, Lorenz, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, let's go ahead and get started. So your first question, Lorenz, is, to get the PBMCs, are you using an initial source of material, an enriched leukocyte sample such as leukopaks, or are you isolating them directly, centrifuging whole blood?
Yeah, thank you, Michelle. So this is indeed a very, very good question, actually. So the MACSQuant PBMC isolation kit has been optimized and originally developed for the fast and untouched isolation of PBMCs from small, freshly drawn, anticoagulated whole blood samples. And by small, I mean in the range of 1 to 8 ml. And the good thing is that with this kit, you don't need any gradient density centrifugation. So you can directly go ahead and isolate the cells from whole blood. Yes.
Okay, great. Thank you. All right, next question. How do you achieve efficient stimulation of CD8 T cells with your peptivators?
Yeah, so this is also a very good question that we often get. So for the analysis of antigen-specific activation, be it CD4 or CD8 positive T cells, you always need antigen-presenting cells that will actually present the peptide. So in this case, the peptivator. So also for CD8, and although this is scientifically not completely known yet how it actually works, if you co-culture T cells and antigen-presenting cells and then spike in the peptivator, the APCs will present the antigen and then activate the CD8 positive T cells as well. And this works very well for intracellular antigens as well.
Okay, great, thank you. Now, are there similar kits available for mouse tissues? For example, enrichment kits for activated T cells?
Yeah, so this is something we are currently working on to say. However, what we already have is we have an enrichment kit for CD154 positive T cells, which is the CD40 ligand, mostly to enrich antigen-activated CD4 positive T cells. For human samples, we in addition have the CD137 enrichment kit, which we at the moment do not have for mouse samples. However, you can always go ahead and isolate CD4 T cells with the CD4 microbeads, and there we have them both for human and for murine samples, or of course with the CD8 positive T cells with the CD8 microbeads, and they are also available for human and mouse T cells.
Okay. All right, next question. Let's see. Okay, this user says, thank you very much for your presentation. Can we carry out the in vivo stimulation of these rare cells through vaccination?
Yeah, I'm not 100% sure how I understand this question, but I will just try and answer. So of course, the especially the SARS-CoV-2 T cell analysis kits for PBMC samples will detect antigen-activated T cells from vaccinated donors. So if you have a vaccinated patient and then you isolate, you draw blood at some point after the vaccination, you should be able to analyze the reactivity of the donor T cells with the components that are included in the kit. And here, what is really nice is that we provide a really pre-optimized flow panel consisting of 8 flow antibodies to help you standardize your analysis. And this is especially important, I think, for the future analysis of T cells, especially in the context of analyzing vaccine samples from vaccinated donors.
Okay, great. What is the difference between the different spike protein peptivators?
Yes, so this is also a very good question that we often get. So I can try and entangle that a bit for you. So first of all these different spike protein peptivators cover different parts of the spike glycoprotein. So for example, the peptivator protein S covers all the predicted immunodominant domains of the spike protein. The protein S1 covers only the N-terminal S1 domain. The peptivator protein S plus covers a part of the C-terminal S2 domain of the spike protein. And if you now would go ahead and analyze the vaccine response, for example, you would actually combine those peptivators in a stimulation mix, and then go ahead and stimulate your sample, your T cells. However, we will also release other versions of the spike protein peptivator that we are currently working on.
Okay, great. Thank you, Lorenz. We have several more questions for you. Will you release peptide pools for the new SARS-CoV-2 mutation variants?
Yes, in fact, we already released 3 peptivator peptide pools for the mutations that were originally detected in the UK and also the ones in South Africa. So the B117 mutation and the B1351 mutation. And it's really nice. So for these mutation pools, we actually offer 2 different peptide pools per variant. So one is the mutation pool, which covers the sequence of the mutated protein, and the other is the respective control pool, which covers the Wuhan wild type pool. And by a separate simulation with this mutation and this reference pool, you're then able to detect and compare these mutation-specific T cell responses.
All right, thank you. So moving on to your next question, when will the whole blood kits be available for analysis of SARS-CoV-2 reactive T cells?
Yeah, also this is a very nice question. So actually we already released an optimized version for whole blood samples early in March. So you should be able to find it on the website. And it's basically the same workflow as for the PBMC kit, but it also includes other reagents that are needed to isolate and then stimulate those cells from whole blood samples.
Thank you. Have you tried to culture enriched activated T cells?
Yes, this is what we are currently actively working on, and we will hopefully very soon release protocols, especially for the cultivation of enriched activated T cells. But of course, I am happy, I think also in the slide deck, there is already one protocol for the expansion of antigen-specific T cells before stimulation, if I'm not mistaken, but this I would have to look up. But I'm very happy to come back to you on a different occasion.
Thank you, Lorenz. Okay, we have time for a few more questions here. Let's see, how many samples can be analyzed using the SARS-CoV-2 T cell analysis kits for PBMC samples?
Okay, so this is a rather easy one. So the kit is optimized to analyze 100 samples. However, for the analysis of, for example, one donor sample, we always strongly recommend to have a separate negative and positive control. So basically 3 samples per donor. So which then means that with one kit, you could analyze 33 patients and then always have a stimulation, a positive and a negative control per patient.
Great, thank you. Is the antibody panel included in the SARS-CoV-2 T cell analysis kit flexible enough to include own markers of interest?
Yeah, definitely. So actually intentionally in the PBMC version of the kit, we left one position, so the PE-Bio 615, which is the PE-Cy5 channel free to include your own antibody of choice. And here, of course, most relevant to analyze T cell activation are, for example, markers such as CD25, CD69, or other cytokines like, for example, interleukin-2.
Great, thank you. It looks like we have one more question here for you. Do you need to activate APCs for sufficient antigen-specific activation of T cells?
Actually not. So in the protocol that is included in the kit completely works out an additional stimulation of antigen-presenting cells. However, of course, you can go ahead and also include, for example, anti-CD28 in your stimulation to further boost antigen-presenting cells and thereby also boost the reactivity you will measure from the T cells. But for the kit's workflow, you do not need to separate the APCs additionally.
Okay, great. Thank you, Lorenz. Do you have any final comments for our audience before we wrap up today?
No, I'm just really grateful to get the opportunity to present you this data. And of course, I mean, one thing I can say is that COVID-19 is a very fast-moving topic. And we at Miltenyi are always trying to stay on top. So we are currently developing many more products for the analysis of SARS-CoV-2 specific immune cells, especially also SARS-CoV-2 specific B cells. And there will be a webinar coming up very soon. And yeah, maybe you just keep your eyes open. And other than that, just stay safe and stay healthy. Thank you very much.
Thank you again, Lorenz, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. If you would like more information on the whole blood analysis kit, please visit their website here in front of you. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care and goodbye.